Hello, I'm Hans Peter and I'm a fermentation specialist here at Aquaporin. Fermentation has been used to produce alcohol and the leavening of bread for ages. The Teutons in Germany already made fluid bread. But the earliest recorded examples of the making of beers come from ancient civilizations more than 5,000 years ago. It was not until the 19th century that Louis Pasteur discovered that alcohol formation was connected to a microorganism, yeast, and he called it fermentation. Later, the Buchner brothers found that sugar could be converted into alcohol by components extracted from yeast, and they discovered the network of biochemical reactions called the metabolic pathways. Advances in biochemistry and biotechnology were further accelerated by the discovery of the genetic code in the 1950s, which led to genetic editing techniques that could be used then to engineer the microorganisms to produce molecules and various proteins. They become little cell factories that can produce many beneficial components, like food supplements, medicines, vaccines, and proteins. Fermentation-based production has advantages over conventional production processes in reducing environmental impact and producing large, steady production volume. Also, it is considered to be a more natural production process, as it has long been used for the production of wine, cheese, dairy products, beer, and alcohol. For example, insulin, which for long was sourced from cattle and pigs and caused allergic reactions, is now actually being produced by a fermentation process. And here at Aquaporin, we produce a membrane protein for the enhancement of water filtration properties of water filters. Hence, many yeasts and bacteria are contributing to our health and a sound environment. But how does the fermentation process work in practice? Yeast and bacteria, like us, need food and oxygen to grow and thrive. All ingredients are mixed into a vessel with water, shortly heated to sterilize the growth medium by inactivating unwanted microorganisms. The microorganism is then added to the growth medium. In the process, oxygen is supplied to the medium and the liquid is mixed with a fast rotating stirrer. Temperature is monitored and controlled. Now something fascinating is happening. One cell of the microorganism is producing a baby cell. That baby cell can produce a new baby cell. So then we go from 2 cells to 4 cells, and from 4 cells to 8 cells, and so forth. Now you may ask, why is it important to grow so many cells? Because that is not the final product, isn't it? Well, whilst the cell produces a new baby cell, it can also produce the compound. So more cells mean more cell factories, which lead to more product. The speed at which a baby cell is produced is called the growth rate. The faster the growth rate, the faster the fermentation process. But sometimes the growth rate outpaces the oxygen supply. The cell will suffocate and they get sick. This is like how our muscles cramp when we run too fast, too long. They produce an acid and we need to stop. Similarly, when stressed, microorganisms also produce an unwanted byproduct, halting their growth and stopping the production. This issue is even more pronounced in engineered microorganisms, which can easily become unmanageable, which drives the fermentation engineer to despair sometimes. The key is then to control the food supply to match it with the air supply. That prevents unwanted byproducts and ensures that most part of the food is being used to produce the product. And so the fermentation engineer will need to steer the fermentation close to the wind, but still keeping it into calm waters. Only then it will lead to the most cost-efficient production process and thereby unleashing the beneficial aspects of fermentation-based production processes to the world, like we do at Aquaporin.